Hello, and welcome to the first joint webinar presented by SEKISUI Xenotech and the Drug Development Solutions Center. My name is Jolanta Golitz, and I'm a liaison between SEKISUI Xenotech customers and services provided by Drug Development Solutions Center. Our facilities are located in Tokai, Japan, where we perform studies every year for clients interested in vivo ADME PK or radiolabeled compound synthesis services. We also offer some in vitro studies related to pharmacology and toxicology. Today, I'm happy to introduce our speaker, Miki Fujishima, who will walk you through several hepatotoxicity offerings we have available. Miki's role is to help clients plan contract studies with the Drug Development Solutions Center to properly assess their drug hepatotoxicity and meet regulatory expectations. Miki received her master's degree in drug metabolism and molecular toxicology from Tokyo University of Pharmacy and Life Sciences in 2007, and later that year joined the team in Tokai, helping us to establish drug transporter study offerings performed at both the Drug Development Solutions Center and SEKISUI Xenotech. She works closely with research and development scientists to develop and improve assays relating to drug-induced liver injury, and also has specialized experience in DMPK test systems, including hepatocytes and subcellular fractions. Miki, thank you for joining us.
Thank you, Yolanta. Today, I'd like to introduce the in vitro study options available to investigate the compound's potential to cause drug-induced liver injury. Specifically for cholestasis, we have a functional assay to determine bile acid-dependent hepatotoxicity and ROC analysis to compare test compound to non-clinical incidence of hepatotoxicity. We also have a short or long-term mitochondrial toxicity study. First, I'd like to provide a brief overview of drug-induced liver injury, or DILI. Precise prediction of drug-induced liver injury through non-clinical studies is very difficult, since many mechanisms are complexly intertwined. Therefore, in a drug's development, comprehensive evaluation from several studies is conducted to predict DILI. I'd like to show two examples. The figure on the left is from AstraZeneca in 2012. They did comprehensive evaluation of drug transporters, BSEP and MRP2, which shows transporter inhibition causing cholestasis from bile acid buildup. They also performed the mitochondrial toxicity test using hepatitis cells and a metabolite-dependent toxicity study and a covalent binding study. The results from all these studies can be looked at together to produce a score, which is part of comprehensive hazard analysis represented by a green bubble. The white figure is from Chiba University in 2020. In this article, they conduct 4 in vitro assays: mitochondrial toxicity, cholestasis, lipid accumulation, and bile canalicula extension, which is not usually conducted, but in this case was appropriate. Mitochondrial toxicity and cholestasis are the most important assays for the prediction of DILI, which is why we are taking about them together in today's webinar. Not only these two assays important for the examples here, there are many articles that conduct these two assays for comprehensive evaluation. I also would like to tell the importance of mitochondrial toxicity and cholestasis from the side of clinical information. In 2014, Pfizer Group investigated 72 marketed drugs of different disease statuses correlated with compound BSEP inhibition and mitochondrial toxicity. The left pie chart shows 9 drugs with severe response and other results were withdrawn or given black box warning. As the chart goes from left to right, the levels get safer. Warning and precaution, adverse reaction, and no mention. The black shade is strong BCEP inhibition and mitochondrial toxicity. Dark gray is moderate BCEP inhibition and mitochondrial toxicity. Light gray is only BCEP inhibition, and white is neither. All of 9 drugs in the left pie chart has BCEP inhibition and or mitochondrial toxicity. As you can see, there is strong correlation between severity risk and the combination of BCEP inhibition and mitochondrial toxicity. These 2 studies can be an important part of drug safety program to predict DILI risk. Because of the importance of these data for drug developers to establish safety of their compounds, the Research and Development team at the Drug Development Solution Center introduced studies to predict cholestasis and mitochondrial toxicity. First, let's talk about cholestatic DILI risk assessment. We offer 2 studies. One is ROC analysis using LDH release and ALB data as a screen for clinical hepatotoxicity endpoint, and the other is a functional assay to evaluate bile acid-dependent hepatotoxicity. Let's first talk about the mechanism of this assay system. As you know, the bile salt export pump or BSEP is one of the drug transporters involved in bile acid excretion. Bile acid accumulation in liver can occur when BSEP is inhibited by drugs. This bile acid accumulation is toxic and can be very dangerous. This time, we have established an in vitro test system. It mimics this phenomenon to assess a drug-induced cholestatic liver injury. Most clients conduct this study as part of a screening to assess the risk in drug discovery. However, if a sponsor sees BSEP inhibition in definitive preclinical studies, they can follow up with this functional assay to see if the bile acid accumulation causes toxicity. The assay we offer is patent licensed from Chiba University. We use sandwich cultured hepatocytes because monoculture don't have bile canonical-like structure to enable the function of bile acid excretion. We also qualify the lot of hepatocytes that we get from SEKISUI genotype for adequate BSEP activity before we use them in this assay. So we know they will show excretion of bile acids if the BSEP is not inhibited. Hepatocytes are seated onto collagen-coated plates and overlaid with material. When the hepatocytes are sandwiched by collagen and material, bile canonical-like structure is formed between the cells. Next, the culture is exposed to bile acid and test compound. In our body, bile acids that imported to cells are excreted to bile canonical by transporters like BSEP. BSEP inhibition alone doesn't necessarily mean there will be bile acid buildup though. Sometimes the body has mechanisms to compensate and export bile if BSEP cannot, which is why we look at representative test system to see if BSEP inhibition does result in bile acid accumulation and toxicity. vesicles are the most popular test system for efflux drug transporter studies and are good for screening whether or not the test article is a substrate or inhibitor of ABC transporters. However, for our purposes, where we need to look at the hepatocyte function when treated with test article, vesicles are not an appropriate test system. Vesicles don't have metabolic activity or regulate the expression level of transporters and enzymes, which are important to the outcome of a cholestatic hepatotoxicity assay. As I mentioned before, plated monoculture hepatocytes are also unsuitable because they don't form the bile-connected-like structure sandwich culture hepatocytes do, and therefore would not have a way to pump the bile acid out. Even with normal BCEP activity levels. So this is why we use sandwich cultured hepatocytes as our test system in this assay. This figure shows the schedule of the study. Day 1 is cell seeding. Next day 2 is maturity overlay. And day 3 is medium change. Bile acids and test article are added on day 4. After the 1-day incubation, on day 5, LDH measurement is conducted as the toxicity assay. Because the formation of bile connectivity is important for the BCEP to pump out bile acid, we want to look at that to make sure the sandwich culture has enough function to observe functional bile acid export or cholestasis. This photo is a sandwich cultured hepatocyte on day 5. In this point, we added CD-FDA to the culture medium. CD-FDA is metabolized to a fluorescent compound CDF in hepatocytes. CDF is excreted into bile-connected-like structure by transporters. So when we see fluorescence, like you see here, it tells us that the transporters are working and the experiment can be trusted to show bile acid-dependent toxicity. I'd like to show the validation data of positive and negative control. We use cyclosporin A and flutamide as a positive control and rosuvastatin and fexofenazine as a negative control. The cryopreserved pletable human hepatocyte lot we use is from SEKISUI Genotech. Orange squares are co-incubation with bile acids, and blue is without bile acids. Positive controls are negative cytotoxicity without bile acid. However, adding bile acid, concentration-dependent cytotoxicity was clearly observed. Negative controls are both negative with and without bile acids. The data is not shown in this presentation, but we also conducted the same test using other hepatocyte lots to check the lot-to-lot variation. Same manner was observed. Today, I'd like to introduce two examples of study design we can offer. First is ROC analysis. We use 24 compounds for which we have clinical information. Using these compounds, we conduct the in vitro assay I just walked you through and calculate cutoff LDH values. Then we evaluate your compound's toxicity by comparing your compound's LDH release value. I'll explain the detail in next slide. The second option is an evaluation of cholestasis. In this study, we conduct concentration-dependent toxicity tests with and without bile acid to get corresponding EC50 values. I'll explain the detail later. In the next few slides, I'd like to explain more detail of the ROC or receiver-operated characteristic analysis. This analysis uses in vitro data and clinical data to plot the curve using LDH release and alkaline phosphatase ALP increase to establish a hepatotoxicity threshold. ALP is a clinical biomarker that indicates disruption in bile homeostasis and/or bile duct injury. More than 1% increase is considered as liver toxicity. So here, we put ALP on the x-axis and LDH release data from in vitro study on the y-axis. From these sample data, ROC analysis shows we get a cutoff value as 18.5%. This means a compound would be predicted to cause liver toxicity if the result of the in vitro LDH assay is over 18.5%. We conduct the same study to get the cutoff value in 3 different hepatocyte lots to account for inter-individual differences in metabolism and drug transporter activity. Two assumptions can be made from this data. One, the presence of bile acid is associated with higher LDH release, so by adding bile acid, higher toxicity levels can be detected. Also, this data shows very similar toxicity profiles among the 3 lots we tested, meaning lot-to-lot variation is relatively low. Using the bile acid plus data from previous slide, we conducted ROC analysis and got each cutoff value. We got the cutoff values around 10% among these 3 lots we tested. It's not shown here, but we also have data from 2 more hepatocyte lots. So we typically have 5 lots data. We also check day-to-day reproducibility and determined there was no significant difference. In addition to the ROC analysis, we also provide a functional assay to estimate risk of cholestasis. We evaluate the cholestatic DILI risk of test article by comparing the toxicity in 2 conditions with bile acid and without bile acid. If you can see the stronger toxicity with bile acid than without bile acid, it says that the test article has a risk of cholestasis. The standard study design is here. We typically include 7 concentrations of test article, and incubation is conducted with 2 conditions, with or without bile acid. Let's look at some example data from this type of study. We used cyclosporin A and flutamide as they are both known to cause cholestatic hepatotoxicity. We measure these compounds' concentration-dependent toxicity with and without bile acids. As you see, the clear differences between these 2 conditions, it means that these compounds would cause cholestasis. From these studies, we can help someone who observes hepatotoxicity in an in vivo animal test to explain observations or For someone who observed strong BCEP inhibition in an in vitro drug transporter study to predict hepatotoxicity in vivo, it should be noted that we use human clinical data and human test systems for these assays, not animal. Next, I'd like to introduce our study to predict mitochondrial toxicity, another important mechanism of hepatotoxicity. Let's start with an overview of test system. This study utilized the scrutiny effect. In vivo, hepatocytes produce ATP by aerobic respiration. However, when immortalized hepatocytes are cultured with glucose-rich medium, which is normally used, ATP production occurs mainly by glycolysis, meaning not by mitochondria. By changing culture of glucose-rich medium to galactose-rich medium, the cells revert back to aerobic respiration using mitochondria. This means that detecting mitochondrial dysfunction in glucose-rich medium is difficult because its normal product, ATP, is produced mainly by glycolysis. So ATP levels wouldn't tell you anything about the mitochondrial function. But comparing to galactose-rich medium, which does make a cell use mitochondria to produce ATP, we can see whether there is a reduction in ATP due to mitochondrial dysfunction. This figure shows the mechanism of mitochondrial toxicity. There are many mechanisms, but the test system today we introduce can be detect almost all of them. One of the distinction of our test system, we can offer long-term evaluation up to 14 days. We can detect mechanisms that cause cell toxicity from longer exposure to a drug. For example, liposomal inhibition and inhibition of mitochondrial DNA synthesis. This slide shows a timeline of the experiment. At first, we culture Hep2 cells with glucose medium in flask. Then we seal cells to 96 well plate with glucose or galactose-rich medium. The next step, we expose to the test article and incubate 24 hours, then conduct ATP measurement. In the case of a short exposure assessment, when conducting long-term exposure assessment, we culture cells with each medium and test article for 6 to 14 days. The data I'll show later. We expose cells 10 days. So here, I share results from short-term exposure assessment. We use rotenone and antimyosin as a respiratory chain inhibitors, FCCP as an coupler, and oligomyosin as an ATP synthesis inhibitor. After one-day exposure of these compounds, the ATP content was measured. Compounds with galactose medium show strong cytotoxicity. After one-day exposure of these compounds, the ATP content was measured. Compounds with galactose medium show stronger toxicity than those of glucose medium. We also tested the toxicity of imipramine and digitonin. They are known as potentially causing cytotoxicity, but not mitochondrial toxicity. These compounds show the toxicity, but there is no difference between glucose and galactose medium. This slide shows a result of long-term exposure. In this study, we use linezolid and chloramphenicol and ATP measurement conducted at 1 day and 10 days. Upper graphs are 1-day exposure of this compound, and the bottoms are 10 days exposure. The toxicity difference of this compound is difficult to see at 1 day. However, the difference is clearly observed at 10 days exposure. In summary, while there is not one definitive predictive assay or methodology to predict DILI, there are some good in vitro assays you can use or to explore certain mechanisms and use comprehensively to predict clinical risk. Drug-induced liver cholestasis and mitochondrial toxicity are two particularly important mechanisms of DILI. We can offer contract services of these two in vitro studies. Thank you for attending our webinar with SEKISUI Genotech. If you have questions about the assay I presented today or would like to inquire about booking a study, please use the Contact Us form on the right-hand side of the top navigation bar, and a service specialist will be in touch soon.